Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I'm very delighted to be here at this important event, SMIC, Science and Mathematics International Conference 2022. I am Sudaryanti, student of postgraduate of Biology Education, Jakarta State University. Let me present my paper with the title is Characterization of Amylolytic Bacteria Associated with Algae at Sepanjang Beach. Enzymes are important components in the body of living things. Enzymes are indispensable for the effectiveness of the completion of a chemical reaction. Amylase is an enzyme commonly found in microorganisms. Amylase enzyme can be used for the food and drugs industries, textile industries, and etc. Amylase enzymes are more desirable because they are environmentally friendly. The breakdown that occurs is more specific and does not cause distorted taste in the final product. Amylolytic bacteria can be found on substrates that produce starch. So in this research, they were also found. If we are able to utilize bacteria that produce amylolytic enzyme, we will get many benefits. Here, the aim of the research. The aim of the research is to characterize amylolytic bacteria associated with algae at Sepanjang Beach. And the slide is the first. We are making artificial seawater and marine agar. And the second is sampling. Algae sampling was carried out on the beach at low tide. Next, the algae samples were put into a plastic sample and placed into cold box for further isolation of bacteria associated with the algae. And the next is isolation of bacteria. Algae samples that have been taken are then rinsed using sterile distillated water. Weigh the sample for 1 gram and then put it in a plastic bag that already contains 9 milliliters of physiological solution. And the last step is amylolytic activity test and bacterial morphology and cell characterization. Okay. After isolation, then it was diluted serially to 10 to the power of 4, then 100 milliliter of the diluted sample was taken and it was put on natural media and sprayed using L glasses. The sample was then incubated at room temperature with the agar on top. The growing bacterial colonies were counted and purified based on the shape, color, and size of the colony. Every single colony was isolated into tiled TSA media for the further testing. Yeah. And here the results and discussion. First is colony morphology. A1 is a code for bacteria that isolated from Cetamorpha-linum. A2 is bacteria that isolated from Enteromorpha intestinalis. A3 is bacteria that isolated from Euchema spinosum. Yeah, you can see that there are so many various of shapes, margin, elevation, size, pigmentation, and optical property of every single isolated. For example, for A1.1 and A16A and A1.10, that bacteria that isolated from Cetamorpha-linum, they have shapes that are circular, while the margin is entire, elevation is convex, size is small, pigmentation is white, and optical property is opaque. Meanwhile, for A1.2, the bacteria that also isolated from Chaetamorpha. You can see that the shapes is spindle, margin is entire, elevation is convex, size is small, pigmentation is white, and optical property is opaque, and etc. There are various shapes, margin, elevation, size, pigmentation, and optical property, like in the table. And here's the cell morphology for all isolated. We can see in this table that there are two types of gram of the bacteria that we are able to isolate it. There are positive gram and negative gram. There are 12 bacteria that have positive gram, and there are 14 that isolated have gram negative or negative gram. For the gram positive, like I said, there are 12 isolated that have gram positive. Seven of them there are have shapes coccus, and 5 is rods. Meanwhile, for the negative gram of the isolated, 3 is cocky, and rods is 11. And here is the amylolytic index test. Yeah. From the result of our calculation, the amylolytic index data of the bacterial isolated we obtain range from 1.02 until 6.03. The amylolytic index were then tested with ANOVA statistic to determine whether the differences in amylolytic index were significantly different or not. The result of ANOVA showed in the table that F counted start hydrolysis index value is higher than F table 5% with a significant level of P less than 0.01, so that there was an effect of different amylolytic bacteria isolated on the amylolytic index value. The data was then tested by Duncan test with a significant level of 5% to determine which bacterial isolated that has the highest amylolytic index value. From the table, you can see that isolated that the highest amylolytic index was we can see that A2.8. So of the our several steps, we know that the highest level is actually isolated that has name or code that A2.8. Okay, and here the image of the colony and clear zone diameter. A is colony diameter with has red line. Meanwhile, B is the clear zone diameter with yellow line. And here the conclusion of our research. First is there are 26 isolated were obtained consisting of 9 isolated of algae samples, 1 or Chaetamorpha lino, and 9 isolates of algae samples, 2 Enteromorpha intestinalis, and 8 isolates of algae samples, 3 Echema spinosum. The second conclusion is the amylolytic index of the bacterial isolates obtained range from 1.02 until 6.03. based on the result of Duncan's further test showed that isolate A2.8 had the highest amylolytic index. And that's all my speech. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.